Okay, so first I'd like to thank the organizers for the opportunity to present our work, and also thank my co-authors, especially Teppo Niinimäki, who did most of the work. So a lot of this track is about precision medicine, which essentially is about building models to help diagnosis and treatment planning for patients using individual data for those patients. And this kind of work has a potential privacy risk because we need to use actual real patient data. So there's been some work basically attacking this kind of data and this kind of models in the past, demonstrating these kind of risks. So these include, for instance, de-anonymizing genomes. So basically identifying, giving names to some published genomes using publicly available data. Identifying participants from some published studies. So basically telling which individuals were part of the case group and which individuals were part of the control group in some study, for example. And finally, so this is by the way called membership inference, which is a sort of fairly common type of attack considered here. And finally, the question we are interested in, which has to do with recovering the training data used to train a model from the published model, which is called a model inversion attack. And sort of final additional challenge here is that all of these have been done for not just in medicine, but what makes things difficult in medicine is that often we are dealing with genomic data, and here the privacy risks are even more difficult because genomes are permanent, irrevocable. Any disclosures also affect relatives, some of which may not even have been born yet. And we don't really understand what you can do with this kind of data. So there is a good reason to be cautious. So here is an example of the model inversion attack. It's not a very easy figure. This was taken from this paper by Fredrickson et al., which basically shows that. So looking at the right end of the curve, which has to do with trying to infer these genotypes from personalized warfarin. So it's a drug dosing model that uses some genotype information for a few genes. So in case they don't have privacy protection or very weak privacy protection, they can reach fairly high accuracy in inferring these attributes or genotypes. And what's also alarming is that they compare this with making these inferences for training data and validation data, and the accuracy tends to be higher for the training data. So the people who contribute to the training data set actually their privacy gets exposed due to that. Now the way to deal with that is, of course, to invoke something that can deal with protect the privacy. Here they use differential privacy. I'll be talking more about that in a minute. But basically it shows that there's a privacy budget that you can tune, and if you tune it tight enough, then you can essentially eliminate this kind of attacks. So what is differential privacy? The concept has recently become fairly popular, and it's used among others by the many big cell phone companies. So Google, Apple, and the like big tech companies. And it's based on the idea of using two what's called neighboring data sets that differ by a single data of a single individual. And the differential privacy criterion basically says that the results of running an algorithm on these two neighboring data sets should be similar, where there's a parameter epsilon that tells how similar they should be. And effectively this then gives any participant in the data set kind of plausible deniability. The results would have been the same even if my data was substituted by someone else's. Therefore, you can't learn too much about what my specific data was. This is a very powerful privacy concept because it provides protection also against adversaries with side information. So many of the attacks in the previously were based on combining the released information with some publicly available information. Differential privacy can deal with that too. It has nice properties for developing algorithms, so that it's invariant to post-processing. Also degrades gracefully under repeated use and composition, so this sort of doesn't break completely. So to get an idea of what it means, this is not an algorithm we're using, but just for an illustration. It's the simplest possible differential private algorithm, randomized response, which can be used to collect answers to potentially embarrassing questions, such as, are you using illegal drugs? And the protocol works such that the respondents are instructed to answer as follows. So they first flip a coin, and then depending on the outcome of that coin, they either respond truthfully or then flip a second coin and then answer whatever that second coin tells. So this gives sort of the intuition behind differential privacy. There is a probability that they will answer truthfully, but there's still this probability that it will just give some noise, and the plausible deniability that even if they answer yes, they can say, well, it was just because of the coin. I'm not really using drugs. You can't really tell me. But when we do the analysis, of course, there is some information that gets retained. But with this, we can show that this is epsilon differential private, and the privacy parameter epsilon here will be of the order 1. And this is important because this is also the level. So this gives an intuition of the level of privacy protection you get from the methods that we are presenting here for much more complex cases. So the specific case that we are looking at is drug sensitivity prediction, where the task is to predict the sensitivity of specific cell lines to cancer drugs using gene expression data. And we do this using data from the Genomics of Drug Sensitivity in Cancer project, using Bayesian linear regression. And in order to privatize this linear regression, we basically add a bit of Gaussian noise to different parts, or sorry, Laplace noise actually here. You can do it either way. So this was previous work published last year. And so for the evaluation, we used the same that was used in the DREAM challenge so basically we look at the ranking of cell lines for each drug and compare the accuracy for that. And the headline result for our previous result was that we managed to develop a method that was significantly, the one in red here, which is significantly better than any previous differential private method. And we could obtain similar accuracy as a non-private method using roughly 4 times more data in this case. So there is a cost to this, but on the other hand, we do can avoid these kind of model inversion attacks. And one of the big sort of maybe not so great points about this particular work is that in order for this to work, we needed to reduce the dimensionality a lot. And so for this previous work, we used prior knowledge of most important cancer genes, ranked based on the number of observed mutations, and then simply picked, in this case, it was 10 most important cancer genes for this particular prediction. And now, while doing this work, it took us a lot of time to figure out how to make this work. Some things we learned during that is that one thing to note is that this differential privacy guarantee is a worst-case guarantee over all possible data sets. And it's not specific at all to the particular data set that you have. And basically what this means is that if you can somehow reduce the space of possible data sets by eliminating outliers, it can help a lot. Also, I already mentioned that this high dimensionality of gene expression data, tens of thousands, is a big problem. So there is some theory that suggests that for a d-dimensional learning problem, so differential private learning requires approximately d times more samples than non-private learning to get comparable accuracy. So basically means that typically you would want to reduce the dimensionality a lot. And finally, learning complex tasks from scratch tends to be pretty hard because the signal you get from the data is less than usual because of this privacy constraints. So if you can somehow combine this with additional non-private data, that can be really helpful. So motivated by these last 2 points, we decided to try to develop a method that could use additional data to learn a good low-dimensional representation, and then using that to do the final prediction. And the contribution of this work is really this new method for differential private learning that works with limited private data. We only have very few samples with these drug sensitivities available, but then combining it with unlabeled public data. So the process graphically works as follows. We have this public data. We learn a representation, so basically a method that mapping that reduces the dimensionality, somehow hopefully focusing on the important parts. And then we take the private data and we simply apply this mapping there, getting a lower dimensional gene expression matrix, and then we use that to learn the differential private predictor, which we can then publish. And for the data, so for the private data, we use the same GDSC data as before. And for public data, we now use TCGA pan-cancer gene expression data. We don't have the drug sensitivities for that, but that doesn't really matter. We can still use it to learn the representation. One potential challenge here is that one of these data sets comes from cell lines and measured with microarrays. The other one is real patient data measured with RNA-seq. We are dealing with this by doing some quantile renormalization. That seems to work somehow, but probably there is a price for that. So maybe having a better matched data might help the results further. Finally, we also make use of the cancer type information available in the TCGA data to set up an auxiliary learning task on cancer type classification that we use for hyperparameter learning. And here we especially focus on the most difficult cancer type pairs to predict, because some of them are really easy, but there's some pairs that are actually quite hard to predict. And the basic scheme of the optimization is as follows. So we have this big public data set. We split off one pair of cancers that we sort of treat as pseudo-private data that we try to classify. Then we learn the representation with this, what's called pseudo-public data, learn the classification here, test it, repeat this over many times, and basically with that we get these parameters, which basically include some hyperparameters for the models, dimensionalities. There's a few different models that we tried for this. So before getting to the actual prediction for the drug sensitivity, here as a warm-up we have some results for cancer type classification. So again here, this is all done under differential privacy with epsilon equal to 1 using logistic regression. And now what I'm comparing here are different kind of dimensionality reduction methods. The differential private classification works such that if you don't do any dimensionality reduction, the results would basically be complete garbage. It's you need to add so much noise that it doesn't learn anything. So random projection is essentially the sort of baseline, and we can see that by using some of these other representation learning methods like PCA or variational autoencoders, we can get a significant boost here. We can also change the privacy parameter epsilon, and as expected, tighter privacy gives you less accurate predictions. Less tight privacy gives you more accurate predictions. And so for the actual drug sensitivity prediction, so now here the sort of anchor point is the gray one here, which was our previous result from last year using these top 10 most mutated cancer genes. That was the optimal for this particular one. And now here we have compared again. The random projection is actually doing a little bit worse. And then we also compared to a previously published differential private feature selection method that wasn't doing that well. But now we see that both the PCA and the variational autoencoder again give quite significant boost here. And approaching the level of what you can get with a non-private method, especially, I mean, if you reduce the dimensionality there. So that concludes my talk. So takeaways would be that if you're working on these training machine learning models on personal data like health records and releasing those models, you need to be careful because it is possible to go back from these models to the training data, at least to some extent. And differential privacy is a technique that can avoid this. It can provide very strong privacy guarantees. Basically, the best thing I can think of that would be compatible with the European GDPR type requirements for anonymity. But it may limit the utility, especially for more complex tasks. There's still work to be done, so efficient differential private learning requires a new kind of thinking, and I don't think we are fully there yet understanding how to do it. Incorporating non-private data in learning is a big help, and our contribution here was to provide a new method for doing that using some big unlabeled public data. Thanks.
Really interesting use of the technology.
Hi. I have a couple questions, really. So in differential privacy, one of the things that I struggle with is how do you select epsilon? Because that really has no meaning in terms of, like practical implementation. So, like, how do you foresee, like that challenge? And the other thing that I really liked your talk, by the way. The one thing that I was kind of struggling with is when you compare the loss of accuracy against other methods, you're comparing against methods that have been reduced down to a lower dimension. But there might be some loss of accuracy caused due to the projecting down to the latent dimension. So shouldn't you be comparing against like a classifier trained on the original data in a non-private manner?
Okay, to answer the last question first, the blue curve here is actually a non-private lasso that's trained on the original data. So that's we've done that in the previous work. For how to select epsilon, I guess it really depends on how sensitive the data is and kind of what are your. We don't really fully understand how it means. What I like to use is this analog here. So if you use epsilon roughly equal to 1, that gives roughly the same level of privacy as you would have from this. And here you can clearly understand what it means. And you can also sort of extend this algorithm for different levels of epsilon. That will give you some idea. One caveat with that, though, is that for these more complex algorithms, here the interpretation of the epsilon is tight. For more complex algorithms, it might not be.
Time for a very quick question. If not, I'm sure we'd love to catch with you over coffee. Thank you so much for a great presentation.